Global warming is affecting our lives in a variety of ways. One of the causes of global warming is the increasing concentration of CO₂ in the atmosphere. Reducing the level of CO₂ has become a matter of the utmost urgency. Plants fix carbon through photosynthesis, converting CO₂ absorbed from the atmosphere into carbohydrates. It is predicted that land plants absorb about 58% of all CO₂ emissions on the planet. Therefore, if we can increase carbon fixation by land plants, we can reduce the level of CO₂ in the atmosphere. NTT Space Environment and Energy Laboratories is working on using genome editing to enhance the natural ability of land plants to fix carbon. This research now focuses on stomatal opening and closing, and flowering in plants. Plants absorb CO₂ from the atmosphere through tiny holes called stomata, located on the surface of their leaves. We will use genome editing to modify the function of the genes involved in the regulation of stomatal opening and closing to increase the opening width of the stomata. This is expected to increase the plant's capacity to absorb CO₂ and thus boost the quantity of carbon fixation. When the genes that control flowering in plants are activated, hormones that induce flowering are produced in the leaves. This hormone causes flower buds to form at the stem tips, which will later flower and then form seeds. Therefore, we will use genome editing to alter the function of genes that control flowering to impede or delay flowering. This will allow the energy normally expended for flowering and seed formation to be diverted into growth, thereby increasing carbon fixation. In addition to these initiatives, we are working on using genome editing to modify the functions of various genes. In the future, we hope to reduce CO₂ in the atmosphere by enhancing the carbon fixation capacity of trees and crops.